Hello and welcome to the eOrganic webinar on increasing plant and soil biodiversity on organic farmscapes. My name is Alice Formiga and I'm the webinar coordinator for eOrganic. eOrganic is the organic agriculture community of practice with eExtension. We're a community of cooperative extension service personnel, researchers, ag professionals, organic certifiers, and practitioners. You can find our recorded webinars, information about upcoming webinars, as well as articles, videos, and more by going to our website at www.extension.org/organic_production. We're very excited to have Louise Jackson with us as our speaker today. Louise Jackson is a professor and cooperative extension specialist in the Land, Air, and Water Resources Department at UC Davis. Her work focuses on biodiversity, soil ecology, and nutrient and water management. In this presentation, she will examine research results from a case study in California on an organic farm with hedgerows, preservation of a riparian corridor, and tailwater ponds. After the 45-minute presentation, you will have the chance to ask her questions. If you have a question, you can type it into the question box on your screen and hit return. If you can't see the question box, click the small plus sign next to the word question on your control panel to open it up. Now I'm going to hand things over to you, Louise.
What I'm going to be telling you about today is some work we've been doing over the past 5 years on increasing plant and soil biodiversity on organic farms. And the idea of this work is both to show the value of plant and soil biodiversity for various ecological functions, but also to focus our research more at the farmscape and the landscape scale. So my presentation is going to be somewhat of a research results presentation, but I'd be glad to ask and try to answer any practical questions when you all come back on the line for questions at the end. So right now I need Alice to click the next slide for some reason. There we go. We'll go up to the, please go back one. Before setting out, I really want to thank the cooperators and students and postdocs who've been working on this project. The funding for the project came from the USDA Organic Research and Education Initiative and the Kearney Foundation of Soil Science. And I really especially want to thank Bruce Rominger and the Rominger Brothers Farms, who were great cooperators in one of our case studies. Sean Smuckler, who's a PhD student who did a lot of the research, and a whole group of other collaborators who made this interdisciplinary type of work possible. Next slide. So I'm going to start out my talk with talking somewhat about agricultural intensification. Agricultural intensification is a process by which farming systems increasingly increase production and income with a variety of sets of inputs that include more mechanization in fossil fuels, more non-renewable inputs such as inorganic fertilizers and agrochemicals. Often there's more frequent cropping, sizes of fields increase. The farms tend to occupy most of the landscape, and in this process there's a loss of biodiversity. And I just put a picture of conventional farming in our area in Central California, where you can see that for many of the row crops that we grow, very high cash value crops in California, they're so valuable that people just farm right up to the road edges, the riparian corridors. And the question really is, why does biodiversity matter? And what should we be doing to either increase it or not worry about losing it? Next slide. So the main topic of my webinar is on looking at farm edges as places to increase biodiversity, but I'll be talking about the field as well. This is a familiar site in a lot of the really intensively farmed areas in California where a slough, in other words, an original creek channel that's intermittently dry and wet. In California we have a summer dry climate, has been modified. And if you look at the little map from the soil survey, the USDA soil survey on the left, you'll see that this creek channel that once was presumably kind of meandering across the landscape has been nicely configured at a right angle to go along the sides of fields. And in that process of modifying the channel, people have really changed not only the configuration of the channel, but its slope, and farming right up to the edge. Most of the plants growing in the channel are non-native annuals that dry out with the summer drought. You can see them dried there. Now the slough provides an important process for farming, and that's because water is it's a source of irrigation water that's pumped. You can see the tractor and the irrigation pipe along the side. Drainage from the fields goes out into the slough. You can see the drainage pipe there right in the center of the photograph. And so these are very important for irrigation, but we also can think of ways where we might manage the situation a little bit better, and I'll explain why in the rest of the talk. Next slide, please. So in our county, Yolo County, in Central California, fairly near Sacramento, we've had a lot of interest in this idea of bringing farm edges back to life. And here's a handbook. You can get this off the web. And the idea of farmscaping is that we want to look at a farm as more of a landscape and not just farm fields, and look at ways to restore some of the functions of those edges. So the kinds of ways that people have worked on enhancing and restoring non-production areas is with tailwater ponds, hedgerows, riparian tree plantings. And this is promoted by federal, state, and local agencies such as EQIP, our local resource conservation district. And in our area, this is often facilitated by NGOs. One example is Audubon, California in our area, where people from groups such as Audubon actually help farmers do the restoration, look for funding to support it, etc. Next slide. So the slide I just showed you was a rather typical site, but here I'm showing you a channel that's been restored with native perennial grasses and other graminoids. This has been done by Hedgerow Farms. This is the main irrigation conduit in our area. And Hedgerow Farms has taken a large role in thinking about how restoration of a channel's edge can occur. What you see along the immediate side of the waterway are carex, sedges and rushes, native species, some deer grass, some native perennial purple nealgrass, some grindelia. And this restoration is really effective in supporting the edges of the channel. But right now, up to this point, when we were doing these studies, we don't have a lot of idea of other benefits that this kind of restoration can do. Next slide. This picture here is in the spring. The previous picture was in the summer, showing our summer dry environment. And here what you can see is that there's, in addition to the graminoids, the natives planted along the channel edge, there's willows. Right on the other side of the willows is agricultural fields. And so these willows act as a barrier. Once again, we're not quite sure exactly what they do. We know that these native plantings not only increase the vegetation along the side of the channel, but they also provide habitat for things like beneficial insects, for wildlife. And our study was then looking more at what was going on in terms of soil and nutrient benefits from restoration and riparian corridors. Next slide. So why do we want this kind of documentation of effects? Well, for one thing, the organic NOP says that we should be enhancing biodiversity on organic farms. Another reason is that riparian zones are protected under the California Environmental Quality Act, under the EPA, and in fact, the riparian corridors in California before European settlement had the highest species richness in almost all the native plant communities. The reason is these are some of the few areas that have water year-round. So various groups which is the Ag Water Coalition, State Water Resource Control Board, the Habitat Conservation Planning people have really emphasized non-point source pollution as a goal, and we're just really interested in how restoration of biodiversity and function can help some of that. Not only that, if we have more information, rather than just recommendations for management, we hope to increase capacity building and get more of this on the landscape. So our studies have taken place for about 5 years with sampling both in the native habitats around the farm and in the farm fields itself. Next slide. I'm going to be highlighting one case study that we did on an organic tomato and grain farm, and this is run by Rominger Brothers Farms. You can see that the location of the farm is a couple hours east of San Francisco, near a little town called Winters. I'll be showing more details on the farm, but it's highlighted in green. It's about a 50-hectare farm. It's positioned along a slough on the south side. It's been organic for about 15 years. Hedgerows were planted about 12 years before the study began. There's several tailwater ponds for capturing sediment. There's a tailwater return system by which the water in the tail ponds is pumped up to the top of the field for reuse during irrigation. And along that riparian corridor, the trees are at least 50 years old. Next slide. So here's a little bit more on the study site. Fortunate for us and our cooperators help, the study was done on pretty much the entire same soil type across the farm, a Tehama loam. You can see that it has a very deep soil profile. Next slide, or next, click the button. And this is just percent silt and clay across the various habitats in the farm: riparian corridor, hedgerows, south field, north field, tailwater pond, and ditches. And they have very similar sand, silt, and clay content. So the nice thing about this was that we could compare the different habitats for their biodiversity and soil functions without confounding effects of soil type. And I should mention that the work I'm presenting here is in revision on agricultural ecosystems and environment in the Journal of Soil Water Society with Sean Smuckler as the first author. So you can get more information for those when they come out. Next slide. So let's look in more detail at the farm. There's a south field and a north field. Those two segments of the north field are managed together. Along the south side is a riparian corridor, and that's Chickahominy Slough. And then the soil survey picture was taken before the hedgerows were planted. They're scattered along the edges of the field. And then we have tailwater pond at the west end of each field, and the drainage ditches along the west end of each field as well. Next slide. So here's more detail on the farm picture. So there were 6 habitats that were sampled over the 2-year study. And the plots for the 2 years, 2005 and 2006, were randomly distributed by habitat across the farm using GIS coordinates. And they were then set up for sampling year-round to look at a number of different features. I'm not going to list those features here, but I just want to get you an idea of these different habitats by the following slides, and then we'll go into the results next. Next slide. So here's the south irrigated field. You can see that it's furrow irrigated. Tomatoes are in the ground. It's managed organically with newer inputs early in the season. And then surrounding outside of the field is some of the natural vegetation. So this field is right at the edge of the landscape, moving up into dry grassland savanna hills. The irrigation water is brought in from the Central Valley irrigation projects. And it's pumped to the top of the field. And then the field has a slight slope about 1 to 1.5%. So that's a concern for sediment erosion down in drainage ditches at the west end of the field. Next slide. Here's a picture of the drainage ditches with the furrows of the field, tomatoes in them, going into this unlined ditch. The water from the drainage ditches then moves into a sediment trap and a tailwater pond. Next slide. The picture of the tailwater pond here shows its rough dimensions. What I should point out is that sometimes it's full, sometimes it's not, depending on how much runoff there's been, how much water storage is going on. But down there at the east end of the field is a pump that pumps the water up to the top of the field and recycles it. Before the drainage water moves into the tailwater pond, there's a sediment trap, sort of a basin, that is instrumental in recovering some of the sediment before the water moves into the tailwater pond. In this case, the tailwater pond is pretty much just lined with sort of spontaneous weedy vegetation. It's not been restored with native perennials yet. Next. Finally, the riparian corridor is on the south side of the field, and it's got native trees, intermittent water flow, and serves as a sort of buffer between the fields and the waterway. Next slide. The hedgerows are native California shrub species. They're very drought tolerant. That means they don't need irrigation, although they'll need some to get established. And they're scattered along the edges of the field. Next slide. So I'm just going to start out with some of the major benefits of farmscaping at this farm. And what I want to first point out is in terms of sediment capture, the tailwater pond was very effective. So this graph on the left looks at total suspended solids in the water, and it looks at the discharge of the water from the drainage ditches from the north field versus the water in the tailwater pond. And you can see that there was nearly 95% decrease in total suspended solids caused by water pooling up in the tailwater pond. So why is that important? Well, the tailwater pond, if it overflows, will run into the riparian corridor. And what we're trying to avoid is having sediment-rich water move into the riparian corridor. That riparian corridor is water that eventually will make its way downstream and serve as drinking water supplies. And of course, the sediment also is not great for wildlife habitat in the riparian corridor. Another benefit of the farmscaping is carbon storage. So if you look at the graph on the right, what you see is for those 6 different habitats lined from the riparian corridor, hedgerows, fields, and then the irrigation habitats, the amount of carbon, megagrams per hectare, stored in the different habitats. And what you can see is that the soil carbon in the top 0 to 15 and 15 to 30 centimeters, so that's about the top foot, is fairly similar across the habitats, except that it is slightly higher in the riparian corridor. Big difference, though, is that the trees in the riparian corridor contribute significantly to the carbon storage, as do the hedgerow shrubs. These were calculated with allometric equations that are published for the trees based on their diameter and height. And for shrubs, we got our own data on shrub species to calculate the total carbon stored. So if we look at just that carbon storage, we see that that riparian corridor that's been preserved on this farm contributes 18% of the farm carbon. If we look towards a meter depth, it'd be more like 10%, but it's still a significant amount of the farm carbon. The soil in the north and south fields hold most of the carbon, but without that riparian corridor, we'd see a significant decrease on the farm. Next slide. We also measured greenhouse gas emissions on a frequent basis in all the different habitats. And what we found was that carbon dioxide emissions were pretty similar annually in all the habitats. So we've got year 1 in white bars versus year 2 in gray bars. And the importance of this data is to show that annually, even though they have different water regimes which can affect greenhouse gas emissions, the riparian corridor wasn't any higher than the rest of the sites. There's some variation from year to year. For example, the north field was lower in the first year. It didn't have irrigated crops on it. Then it was higher in the second year. But across the board, these are pretty similar values. When we looked at nitrous oxide, which is more potent as a greenhouse gas, what we saw that it is slightly higher in the tailwater pond and the ditches, which are wetter. It's a process of probably mainly denitrification on this farm. But overall, these were very low rates on the farm. The low and inputs on this organic farm, much lower than is typical and conventional, made for low rates of nitrous oxide. So that carbon storage then is pretty much an indication of the ability of the different habitats to form to mitigate climate change. I should mention the tailwater pond and ditches are such a small proportion of the total farm area that their slightly higher rates, which were low to begin with, don't have much effect for the whole farm budget. Next slide. So in terms of soil drainage and leaching, we used anion exchange resin bags buried about 2 feet deep to look at which habitats had the most leaching of nitrate. And what we saw was fairly low rates across the board for all habitats, only maybe about 20 to 40 kilograms of N per hectare. That comes out to be slightly lower than that for pounds and per acre, same general range of units. And what you can see from the data is that our rates were slightly different from year to year, but across the board they were just slightly higher in the ditches. However, when we look at infiltration rates of the soil, they were lower in the tailwater ponds and ditches. That's probably because there's clay particles that are moving down the irrigation sediment movement. And because of that, that might be blocking some of the leaching that occurs in the drainage ditches and keeping those rates fairly low. So the nitrate leaching was not much of a problem on the farm compared to what we see in conventional farms. Dissolved organic carbon in the soil solution was measured with lysimeters. And what we see there is that DOC is slightly high on the farm, especially in the riparian quarter. It gets the leaf litter, and that may contribute to the carbon in the soil solution. Overall, though, the tailwater pond's water is about what's typically seen in tailwater ponds and waterways throughout the region, about 4 to 5 milligrams per liter. So the management on this farm is not contributing significantly to the DOC that might potentially run into the riparian corridor and into the drinking water. Next slide. Biodiversity. I started with the environmental parameters first, but let me now move to some of the different kinds of findings that we got in terms of vegetation, earthworms, nematodes, and microbial diversity. In terms of vegetation cover, we found that the native woody perennials in the riparian corridor and the hedgerows contributed significantly to the biodiversity of native species. The non-native species are the darker type bars, be they legume, grass, or forb species. And what you can see is that there a lot of non-native species, weedy species essentially, across the entire farm. There are not very many native forbs or grasses in the fields or pond or ditch habitats. So the riparian corridor and hedgerows end up being important places for harboring native plant diversity. Earthworms and nematodes were pretty similar in all the habitats. There was one type of earthworm, a megascolectidae, that was dominant throughout all the habitats, slightly less in the ditches, but otherwise pretty similar. Nematodes, in terms of just nematode numbers, were not significantly different across the different habitats except for tailwater ponds and ditches. And what we found was that bacterial feeding nematodes were pretty dominant in most of the situations. Some plant parasite and herbivore nematodes in the non-irrigation habitats. But surprising, very few predators and omnivores, the higher trophic levels, even in the riparian corridor and the hedgerows, which are less disturbed. Also kind of surprising was that the microbial communities is described by phospholipid fatty acid analysis, which looks at the lipids that are in the membranes of the microbes in the soil. Were slightly more abundant in the riparian corridor, but not much. These assays were done in Kate Scow's lab here at UC Davis campus. What we found was that many of the biomarkers were not able to be classified. Those are the white parts of the bars. Fungi were slightly lower in the irrigation habitats in the riparian corridor and the hedgerows. But overall, what we were surprised about from these data was that neither earthworms, nematodes, or microbes were that much different in the riparian corridor and hedgerows. And what we're wondering is, could these habitats have been fairly disturbed in the past and not been recolonized after disturbance? We're not really sure. We're looking into that further. Vegetation diversity was higher in those habitats, but the soil biodiversity is not that different. Next slide. So I'm going to move on then and talk a little bit more about the tailwater pond and runoff. And that's because the tailwater pond is serving a really important function at this farm. As I said, it's not been revegetated with native species, but that is a potential that could occur in the future. So this is somewhat detailed, but what I just want to point out is that we did this different in the 2 years. The south field had tomatoes on it the first year, so it was a sample at the top of the field where the irrigation water was delivered. At the sediment trap, discharge point, and in the tail, that's number 2, and then if you go to the higher resolution drawing in the sediment trap itself. On the second year in the north field, we sampled irrigation ditches shown there is in points 4 and 5, and also in the tailwater ponds, sediment and tailwater ponds in 6 and 7. So I'm just going to go into more detail in that very first graph that I talked about at the beginning. Next slide. So just something about the management on the farm is our main input of nitrogen was through and phosphorus was through composted turkey manure. It's composted with great pumice, and it just gives an idea of the C/N ratio, nitrate, phosphate, potassium. The field was divided up into a side that had a fallow that had quite a few weeds and a mustard cover crop. And I'm not going to go into this in much detail, but I just want to point out that it was a very late season in 2006, and it was hard to incorporate the cover crop, so it ended up with a very high C/N ratio. This ended up having a negative effect on plant yield. But it did, the cover crop did restrict sediment movement into the ditches and tailwater pond. So that there was a trade-off there. I won't go into it in more detail, and I just want to emphasize the various steps in the tailwater pond. Next slide. So what we did was we put these automated samples out at the ditch coming out of the crop field, a drainage ditch, and then in the tailwater pond itself. And these are samplers that can measure both flow rate and take intermittent samples for concentration of constituents. Next slide. So what were the effects on the sediment of the sediment trap in the first year and the tailwater pond in the second year? The reason why the sediment trap was only sampled in 2005 was that the tailwater pond never filled up with much water, so we just measured the sediment trap. And what you can see is that for TSS, total soluble solids, the sediment trap and tailwater pond were really effective in reducing the sediment load in the water. What we saw was that nitrate and dissolved organic carbon went up slightly in the tailwater pond compared to the drainage ditches that were going into them. It might be because of microbial activity, we're not quite sure. But in both cases, the concentrations were nothing alarming. They were quite low. And we're not seeing a lot of movement or a lot of effect on any of problems in the sediment trap and tailwater pond compared to local conventional fields. I'm going to go back a slide, please. And I just want to point out that from this little diagram in this slide, you can see that the dotted line going from the tailwater pond out to that edge where the trees are shown is where if the tailwater pond did overflow, it would go directly into the riparian corridor. Next slide, and then move on to the next one. So the summary from this farmscaping study is that this idea of keeping and restoring riparian habitat and planting hedgerows is important for both carbon storage and plant biodiversity. That having the tailwater pond and the sediment trap was a really effective way to trap sediment. It prevents movement of nitrate and DOC out to the slough, to the riparian corridor, and it enables recycling of water through the return system. So if the pond and trap were not there, we'd see a lot more movement out to the slough, even though the constituents weren't at levels that were particularly high. So what we found on the farm, I haven't talked a lot about water and management in the field, so that was another part of the study. We can say they're really carefully managed, and the actual amounts of leach nitrate and emissions of the greenhouse gas nitrous oxide are quite low. So overall, the farm was carefully managed for organic production, and then the combination of the physical pond and sediment trap interventions and the farmscaping further contributed to environmental quality. Next slide. So overall, what I just want to point out with this next thing, you can just click through these different slides quickly as they're in steps, is that our long-term objective for this type of work is to look at the kinds of policy and market changes that could be incurred with subsidies and premiums for people who might be willing to do more farmscaping. In other words, get some, you can keep clicking those to get them through the different slides. So the idea is that we've looked at how farmscaping makes changes in different kinds of biodiversity, some of the changes in plant and soil community-mediated functions. And what we next need to do is some sort of economic analysis. We can look at what the benefits are in terms of ecosystem services versus the cost and what it might take in terms of subsidies and premiums to get more people on board for this. Next slide. I'm just going to finish up this talk with a bigger scale project that we've been working on, just because that particular study was one farm. And you can see that that farm in particular is the lowest red dot on this greater picture of an aerial photograph of the landscape at large. And so the idea of this study was, okay, what happens when we move across the landscape and look at more different kinds of farms, more different kinds of soils? What do we see along riparian corridors? And so all the sampling points that you can see here were chosen along some sort of a waterway. They were either natural streams, sloughs, canals, and they were picked with a random process with a GIS system. An ecological assessment was made at each one of these points, so they're kind of a representative set of points across the landscape. And then we measured an index, essentially, of agricultural intensification at each one of the sites based on what the farmer used in terms of non-renewable inputs, how much tillage there was, what the landscape complexity was at each site. This on aerial photographs, and I'm just going to give you an overview. Next slide. That's strange. I'm not sure why it blinked back to the, there we go. Okay, so what is to show some of the different kinds of farms that were sampled, everything from grazed grassland to, this is organic walnut orchard along a canal, irrigated row crops, by and large deep soil profiles everywhere. In each case we sampled at 3 points away from the waterway, everything from organic to conventional, grain, vegetable, and orchard, and grazed systems as well. Next slide. And this is just what I wanted to summarize that study by is, so here what we're trying to do is look at the different amounts of richness of microbial communities, nematode communities, and vegetation communities against that agricultural intensification index, which goes from essentially 2, which is very low intensification, say grazed savanna, to high agricultural intensification, which is 14, 15 in the very high input irrigated agriculture. And what you can see here is that for all the different types of biota, there was a decrease with agricultural intensification. All the slopes are significant. The greatest decrease, though, was shown for plants, and that kind of fits what we found with our on-farm case study, is that PLFA, microbial biomarkers, and nematode taxa seem to show less heterogeneity across this landscape than do plants. We're not really sure why. Then if we look at the soil quality parameters, for example, soil nitrate, and these are a log scale, so the numbers don't quite make sense in terms of actual values. I can give you those actual values, but nitrate is very impacted by agricultural intensification going up. So soil phosphate, which I haven't shown here. So carbon goes down with our agricultural intensification index. And then another environmental quality parameter is riparian health. That's a set of visual measurements that we do on the channel width. It's the riffles, the type of vegetation present, the types of pools. That goes way down with agricultural intensification. So overall, what we're seeing is, in terms of biodiversity and these various measurements of ecological functions, is a decrease as we increase agricultural intensification. Next slide. So the conclusions for this talk are, first of all, that if we were to combine native perennials with physically engineered structures such as tailwater ponds and sediment traps, we might be able to even offer more functions than either alone. And that's something we'd like to test in our future case studies on farms. Although there aren't a lot of people doing these things, we're really lucky to have a cooperator who had integrated so many different types of interventions on one farm. We need more landscape and whole farm studies to show the potential for increasing biodiversity and its effects on environmental quality. Oops, it looks like I lost my presentation there. But I did want to say that in terms of organic management, at least in our area, organic farmers seem to be ahead of the curve on some of these types of interventions, and that gradually conventional farmers are coming on board with some of the same practices, and that incentive for biodiversity on organic farms could further help to give information that could increase this across the entire landscape, even though organic farms are not the dominant form of agriculture. Well, it looks like I've lost, I'm not seeing the screen.
You may have minimized it, I'm not sure.
Okay, so I think I'm, let's see. Well, why don't we just start with questions? That's the end of my talk.
Okay, well, thank you very much, Louise. We now have about 20 minutes left over, depending on how many questions we have. For those of you who missed the beginning of the presentation, you can use the question box on your screen to type in questions and hit return. If you can't see the question box or it's closed, you can click on the small plus sign next to the word question to open it up. And we'll be reading the questions out loud, and we'll answer as many as we have time for. This webinar will also be posted on the eOrganic website within the coming week in case you missed anything or you'd like to hear it again. And I've put the URL of our website with all our archived and upcoming webinars on the screen. But if you lose track of that, you can also just Google eOrganic webinars and you'll get the full schedule with links to the upcoming and archived presentations. So we do have a couple of questions coming in. The first one is, the soil biodiversity might change dramatically depending on the season. When were the soil samples for nematodes, fungi, etc.?
Okay, good question. They were sampled in spring of each year. What we did was we wanted to find a time when all habitats had similar soil moisture. That's because our climate is so variable, and the riparian corridor and hedgerow areas will dry out in the summer, whereas the irrigated fields will still be wet. So we picked March to April as a time when temperatures were warm, soil moisture was abundant, and we sampled at that time in both years as the best possible time to go across all the different habitats. And hopefully that also had the highest diversity because the inputs of carbon were still high, and there hadn't been that much disturbance.
Okay, next question. Are you pooling the conventional farms and the organic farms to see if there's any difference between them with respect to the various parameters being used to measure agricultural intensification?
Okay. Well, as it turned out, since we were representatively trying to sample our landscape, we only had about 3 or 4 organic farms of the 20 farms that we sampled. And they were quite different. One was tomatoes, one was walnuts, and one was grains. And so because we did this representative sample, we didn't get enough reps, essentially, to compare organic versus conventional. That's something that would be really interesting to do in the future, but it would take a different sampling regime.
Okay, next question. How did you measure agricultural intensity?
Okay, so what we did was we had about 14 or 15 different yes-no questions about every site. The questions had to do with inorganic fertilizers, tillage, pesticide use. They also had to do with the surrounding vegetation or land use type around every point. So for example, if a given point was surrounded on all sides by intensive agriculture, that would yield a higher score than a farm that was, say, only 20% of its perimeter was surrounded by an intensified type of farming system. So it was a composite of both on-farm management plus the complexity of land use types around the farm point.
Okay, the next question. Are farmers interested in also improving habitat for wildlife or animals, or are they concerned that with the increased focus on food safety, improving wildlife habitat might become a problem for them?
That's a really good question. I should mention that we did a farm survey of about 28 farms. I think that was how many it was, in the same area as the landscape aerial photograph that I showed you. And that's a paper that was published by Sonia Brodt, B-R-O-D-T. And it found, what it suggests is that farmers are planting Some of these hedgerows retaining riparian corridors, because they're concerned about wildlife, they think that it's important, but there's not really that much data supporting that idea. So it was beyond the scope of our study to do the wildlife parameters, but one of the reasons that I have to say that this kind of farmscaping is going on under the auspices of Audubon is that they have shown that this kind of additional native woody perennial species is favorable for bird habitat. In other places in California, such as the Salinas Valley, there's been a huge uproar about removing native vegetation from around farm fields. It's because there's a lot of fresh market vegetables. In our area, we have way less fresh market vegetables. We have tomatoes and other things that are not necessarily harvested fresh market, although there's some melons and zucchini. So in our area, there's just a lot less concern than there about the food safety issues.
Okay, well, thank you very much, Louise, and thank you for all your questions. Again, this webinar will be posted on the eOrganic website at www.extension.org/organic_production, or simply Google eOrganic webinars. And on that website, you can also find out about our upcoming webinars. Thank you again, Louise, and thank you all for coming.
Thank you.